So I think we're going to get started. It's my pleasure today to announce our speaker, Dr. Dennis Steindler, who's going to be giving the 5th annual James Joseph Memorial Lecture in Neuroscience. And Dr. Steindler comes to the HNRCA from the University of Florida, where he was the Joseph Bagnan Shands Professor of Medical Research, and he was a professor in the departments of neuroscience and neurological surgery. And between the years of 2004 and 2010, Dr. Steindler was the Evelyn F. and William L. McKnight Brain Institute director. They do a lot of naming at University of Florida. And we were really excited when Dr. Steindler agreed to come to the HNRCA to take the lead of the neuroscience laboratory. He did his undergraduate at University of Wisconsin-Madison, and he did his doctorate in anatomy and neurobiology from the University of California in San Francisco. He has just an amazing CV. So, and right now what he's been working on is the growth and transplantation of normal brain and cancerous stem cells. He's been working on this for over 25 years. He has published in many prestigious journals, including Lancet and Proceedings of the National Academy of Sciences. So without further ado, I think I'm just going to turn the podium over to Dr. Steindler so he can talk to you about many of his wonderful ideas. So welcome, Dennis.
Thank you, Sarah, for that wonderful introduction. And it's really a treat to be a part of the HNRCA, and I'd like to thank you all for being willing to maybe collaborate, hopefully, with me soon on lots of different things. And what I'll talk about today are the things that we have been working on, including looking at what my collaborators, in particular Brent Reynolds, refer to as functional foods, or nutrients, to begin to attack some of the degenerative diseases and the cancers that we know have many of their roots in inflammation, which I know a lot of the people here study very, very elegantly. So I'm going to talk to you about these things, but I have to begin by sort of giving you a little history of how somebody who's interested in studying how the brain develops, which is what I'm trained to be. As a developmental neurobiologist, I'm supposed to be figuring out how to build a brain. But I got a little bit sidetracked in the late 1990s when we came upon cells in the brains of mice and human beings that just behaved too dynamically, too powerfully to be considered postnatal and adult cells, and we actually got caught up in stem cell biology. So I'll take you on a little history of how somebody who's interested in studying how the brain develops and also interested in how you apply our knowledge of how the brain develops to repairing a brain that's damaged through disease or injury, or in the case of the cancers, when a brain tries to grow too much tissue, how do you deal with that? So the history of our work in stem cell biology was pretty much coincidental with this book which came out from Stephen Hall around the year 2000. And if all of you remember back then, that's when the stem cell debates began. That's when we were seeing on TV talks from presidents and our political leaders saying we have to legislate this field of stem cell biology and regenerative medicine because we actually are working with cells that are not just potent, but are at the very core of what is the definition of life. Where do we get these cells from? Are we taking lives to take cells? And so on. And Stephen Hall's book, he's a science writer, I believe for one of the Baltimore newspapers, published this book called Merchants of Immortality, where he predicted that regenerative medicine and stem cell biology researchers are in essence going to become merchants of immortality. We're going to be offering people the ability to live disease-free with high quality of life for very many, many years if the stem cell promise holds true. Still remains to be determined. So this book sort of set a course for describing a transition that I had in studying how cells and molecules in the young rodent brain and in the young human brain actually set up all of the circuitry arrangements that help build our brains. So this slide can be a review where everybody knows that in the cerebrum of us, we have representations of all of the things we are and all of the things we do. Those are called our brain maps, and they tend to have topia or topic on the back end of a word, like retinotopy for the visual system, and somatotopy for the body, and tonotopy for hearing. Well, I was interested in somatotopy and the representations in the human frontal lobe and parietal lobe of our face and fingers and all these things and how they get built. But the way in which my lab has been studying that for many decades, through my original collaboration from Carol Welt in the early 1970s, where she was recording brain cell activity in the somatosensory cerebrum of rats, and then made little lesions where she recorded from the face or the feet or the tail, and then processed the brain sections in very special ways post-mortemly from these rats, and found out, even without the magic marker here, that there's a map of the rat in the somatosensory and motor cerebral cortex, just like there is a map of us, our homunculi, in the somatosensory and motor cortex. But with rodents, they don't use their fingers and things like we use so much. They use their whiskers to get around in life. And she and Hendrik van der Loos and Tom Woolsey discovered that in the whisker representations of rat, the neurons in those areas line up in little cylinders, each one representing a single whisker. And you can actually see 5 rows of them if you process the cerebral cortex of a rodent in a particular way, where each one of these little squares represents one whisker on the side of a face of a mouse or a rat. So my lab was intrigued by that as a way in which we can study how maps get formed in the brain. So we were looking for cells and molecules that are actually predictors of how to build these maps. And what we came upon was a glial cell that we identified in the 1980s, here seen under the electron microscope, that we called a boundary astrocyte, because it ended up living in the borders between each one of these whiskers before this map exists. Actually, in the first postnatal week of a mouse, this map appears before the brain cells are even aligned and sort of foreshadows or predicts there are going to be 5 rows of whiskers that this rodent has. You have to build 5 rows of neurons in the cerebrum of this mouse or rat, because that mouse and rat can be blind and deaf, and as long as they have whiskers, they can get away from you in a broom if you're chasing them in a kitchen, and they can hunt and eat. So we were studying cells and molecules that astrocytes made that foreshadowed this pattern, and we came upon extracellular matrix molecules, molecules that had glycosylated motifs, sugar motifs, added on top of a protein, a lipid, or a glycosaminoglycan. And those glycoconjugates actually appeared before the neurons were aligned, and there's a critical period in the first week of rodents whereby you can do a little barbering on the side of their face and take off whiskers, and then look in the cerebrum, and we saw our glial cells and our molecules actually opened up when we removed whiskers to produce a boundaryless row of whiskers that allows plasticity. This plasticity critical period was crucial because we didn't know in the 1980s that this astrocyte, which makes all of these molecules, is actually a stem cell. Now, the way in which we came upon it being a stem cell, and the way in which I'm going to take you through this talk, which will hopefully convince you that the reason why we are studying nutrients, dietary supplements, and phytotherapeutics is that we can actually regulate the expression of extracellular matrix molecules by stem cells using these dietary contributions, largely through regulating extracellular matrix and growth factor and cytokine expression, which everyone in this room knows is things that we study in inflammation. So I'm giving you a little warning already. The reason why I'm here and so lucky to be part of this wonderful center is that diet and nutrition and what we are bold enough, I think, to be willing to refer to as phytotherapeutics are the way in which we don't only regulate brain development, but this is the way we're going to regulate brain disease. So these molecules and these glial cells aren't only present in these boundaries around forming units in the cerebrum, they actually go to the germinal matrix of the embryonic and fetal rodent and human brain, where all of our brain cells are always produced around the ventricle system. These are cerebral spinal fluid spinal fluid-filled spaces, in this case, the lateral ventricle. And up in the cerebrum of rodents and humans, this very dense red area here is showing us the expression of molecules referred to as chondroitin sulfate proteoglycans. These are glycosylated GAGs, glycosaminoglycans, with proteins as well that are expressed by stem and progenitor cells and support cells. While we're building the brain, all of our stem and progenitor cells live along the ventricles, then they migrate out, in this case into the basal ganglia, up here on top into the cerebrum, so they foreshadow brain development. But these same molecules go away in the adult rodent and the adult human brain in the cerebrum, in the basal ganglia, where we see them during development, and they sit as a very dense residual layer along the ventricle called the subependymal or subventricular zone. That until we studied this area in the 1980s, we did not know it is the source of stem cells, not only in rodents, but also in the human brain. Our current collaborator, Brent Reynolds, who will be working on a lot of the things we work on here, and hopefully in a very committed way soon, Brent Reynolds actually discovered this neural stem cell in 1992 serendipitously by just culturing the forebrain of adult rodents and postnatal rodents and finding that they make spheres that I'm going to show you in a second called neurospheres that are little colonies of stem and progenitor derived cells. So we didn't know that when we were seeing these patterns by just doing immunolabeling experiments of the developing and the adult brain where we see them present where stem and progenitor cells should be. But then through follow-up studies, through some of my students and postdocs, in particular back in the 90s, a very gifted student who's now a faculty member at Florida State named Eric Laywell, discovered that in the adult brain, you can see that these extracellular matrix molecules are not expressed very much in the standard operating way in brain parenchyma, unless you have, in this case, a penetrating injury, a needle injury, where 3 days after injuring the adult cerebrum or cerebellum of mice, he found that these same astrocytes upregulate their messenger RNAs for these developmentally regulated proteins and produce something that we've all known in restorative neuroscience to be something that we thought was bad, and that's called the glial scar. Now, I don't know if any of you have paid attention to the work in the spinal cord field of why doesn't the spinal cord regenerate when you have a traumatic injury to the cord. And a lot of the spokespeople over the years who've tried to get that research to advance, including Christopher Reeve and others who said, we have to overcome this scar that forms when you make penetrating injuries in the brain, and these astrocytes and their cadre of molecules that they express to cordon off the wound are really bad guys that prevent regeneration. Well, it ends up they're not bad guys. What they're doing is they're secreting these molecules to actually get a subpopulation of astrocytes that Eric discovered in 1992 to actually be a stem cell. So now not in normal brain development only, in mice and humans, but now in the injured brain, these molecules and these same cells are crucial. And last but not least, in this lower panel right here, what we're looking at are brain tumor specimens from a type of brain tumor called a glioma, very, very serious type of neoplasia, in particular a glioblastoma that we thought we should also study to see if these cells are there. And what I'm going to tell you in a few minutes is they're there, but they're not there the way we thought they would be. They're there associated with new blood vessels forming and neovascularization, which I know is a very strong interest of the Neuroscience and Aging Lab for all the right reasons, and will be targets of some of our nutrient therapies as well. But just be warned, I'm going to tell you a lot about injury, disease, and cancer, stem cells, and which cells and molecules are involved. Now, this slide is meant to remind me to tell you that these developmentally regulated extracellular proteins that young stem cell-like astrocytes secrete while the brain is developing in all mammals, and one would presume there is a critical period based on what I told you about plasticity. And I can tell you that postnatal day 15 in the mouse, postnatal day few years in the human, all of these developmentally regulated molecules get downregulated, and you don't see them unless there's an injury or disease or cancer, except for one strange area, the subependymal zone, that in mice and humans always has them present, and we now know always has stem cells, as well as another region of the adult rodent and human brain that we consider to also be neuropoietic, constantly making new brain cells. And thanks for the interests of the Neuroscience and Aging Lab, that's a structure called the hippocampus, which is involved in lifelong learning, memory, and mood. So just know that these molecules disappear in their boundary patterns unless we make injuries. They reappear as reactive glial scar astrocytes that continue to divide, that the field thought were very, very bad cells. But I will convince you in a minute through our work and the work of Magdalena Goertz in Germany that these reactive astrocytes are actually stem cells. So, in summary, what I've been telling you about so far is a diverse set of neurogenic astrocytes that actually we can see during development and in the adult brain to follow a very, very special course of differentiation where we can now look at every different type of astrocyte sitting in the cerebral cortex of a mouse or a human. And they may all look the same under certain types of stains or immunolabeling approaches, but they are all extremely heterogeneous and diverse. So there are many types of them, including this little guy that my friends and relatives refer to as something I drew that looked like Snoopy. This cell stays with a little ciliated process in all of the ventricles up and down from the front of your brain all the way down your spinal cord as a quiet stem cell ready to repair you when needed. But that's the good news. They respond to injury, they upregulate extracellular matrix, they try to do reactive neurogenesis to rebuild the brain after penetrating injuries or neurodegenerative diseases. But these brain marrow regions that live in the cerebrum, called the hippocampus and the subependylar zone, we can culture as stem cell cultures and make colonies, or the neurosphere assay that Brent developed, that make all of the neurons, astrocytes, and oligodendrocytes of the adult brain. But when they make too much brain tissue, that's when we call them brain cancer, and that is also not a really very good thing. So, up until now, I focused only on the stem cells that lived along the ventricle. I'm going to tell you, as well as in the hippocampus, that we also know there's stem cells that live in the cerebrum that one of my students cloned and characterized. That's a really important cell. I'll mention that guy in a second. But you all need to know, for sake of a little history that I'm about to give you, when a guy like me who studies brain development gets caught up in stem cell biology, some good things can happen and some strange things can happen. So first, the good things. We had alternatives to thinking only about fetal or embryonic stem cells as the only sources of very, very plastic cells that can help build injured and diseased brains back into normal standard operating brains. We have adult central nervous system stem cells that live along the ventricular system and act truly like stem cells in cell culture and in vivo experiments. The fetal nervous system has huge numbers of stem cells, but bone marrow happens to be sort of the king or queen of all stem cells in our body. These are the cells that have been transplanted for numerous diseases that we all know about. They have the ability to be very plastic. They're almost as good as umbilical cord blood stem cells. But the idea in neuropoiesis and stem cell biology at the turn of the 1900s and 2000s of this sort of work period was that we have a variety of sources of cells that come from embryos that are controversial, fetuses that are controversial, considering we have to take them from some source. But wouldn't it be nice if adult bone marrow and umbilical cord cells would be the cells that we can use for cellular therapies? So this huge buildup is to tell you something that happened to me that was really rather remarkable. And that was a hematology oncology, a very gifted young investigator named Chris Kogel came to me and said, you know, Dennis, all of the people who work on brain don't respect the bone marrow stem cell enough. And my prediction is that bone marrow stem cells will be not only plastic enough to help a diseased or aging brain get a little bit younger, but they may actually turn into brain cells. Now, I don't know if you all have been paying attention to the aging stem cell press releases coming out recently of some people who are beginning to become interested in having bone marrow transplants from young donors, especially if we're mature like myself, because the idea is through parabiosis experiments being done by the Weiss-Corey lab at Stanford, it's found out that you can take young bone marrow cells and introduce them into an old mouse, and that mouse de-ages. So, fountain of youth. Okay, that's one interesting possibility. The other thing is that as stem cells get older, particularly adult stem cells in bone and brain marrow, maybe they contribute to some of the degenerative diseases that we all very much are interested in this wonderful center, in particular, Alzheimer's disease. So pretty soon, I think we'll all be seeing a paper that comes out from that lab that says you can take Alzheimer's mice and transfuse them with bone marrow from young mice, and they do a lot better on cognitive tests. Okay? Keep that in the back of your mind. Now, Chris Kogel, who's a hematology oncology expert, who came to me and asked me the following question: would you be willing to look at brains of patients who had undergone bone marrow transplants with cells from a family member that we can identify by looking at the X and Y chromosome? In particular, we had access to the brains of patients, in particular breast cancer patients, who had had bone marrow transplants from their brothers. Now, these patients who had wonderful success stories, most of them, were their standard of care chemo and all of the other things for breast cancer that were done, including biologics and radiologics and whatever. Many of them also got bone marrow transplants, but when they got one from a brother who has a Y chromosome, and their cells have X chromosomes, and they promised their brains to us post-mortem in a very, very kind way. We began studying the brains of these patients, and what we found was just earth-shaking, was that up to 5% of the new neurons, astrocytes, and microglia in just the temporal lobes of these women belong to their brothers. So, actual potential transdifferentiation of bone marrow cells into brain cells in these patients. And we had a really interesting time proving that this was real by doing X and Y chromosome fish, because at exactly the same time we were doing this study, my colleagues at the University of Florida discovered that stem cells have the propensity to fuse to adult cells and make so-called hybrid polyploid cells. So if you looked at a cell that had X and Y chromosomes in it, you would have to make sure that there's only one marker for each and that they weren't polyploids by the result of the brothers' cells fusing to already differentiated adult neurons and glia. So we had to prove that quite rigorously for that study, and I can tell you, since my colleagues invented those methods, we felt pretty good about that. But I can tell you that the world of stem cell biology still very much objects to the idea of a bone marrow cell turning into a brain cell. Okay, I had to do this whole build-up because this is the strange and weird thing that happened to me. Okay, now this isn't meant to show you that I know people who might be running for president. Please don't interpret it this way at all. Interpret it as, in 2001, when I moved to the University of Florida, I was the first stem cell researcher recruited to the state of Florida. One day I get a call from the administration saying a car will pick you up in front of the building and take you someplace. Don't ask questions. I ended up getting to go to this guy's office. So I'm standing in Governor Bush's office. It's right around his time his brother is making policy via the Crawford White House speech on how many embryonic stem cell lines will be allowed to be used by nerdy guys like me. So I'm now standing in this office with the governor and his cabinet, and he has a pad of paper and a pen, and he has two questions for me. The first question is, could I define when life begins? Because he's making decisions on which cells should be legal, when we have to not take lives to get them. And the other decision he was making at that time was a very interesting case referring to a patient named Terry Schialvo. Do you all remember that? So the question on when life begins, I'm not going to tell you the joke that I told Dr. Rosenberg. That'll be done another time. But when he asked me to define what is cloning and will I make sure I never do it in one of his universities, I promised him. I said, Governor Bush, no cloning for me, no one I know, don't worry about it. I get back to my lab, and this slide is in my inbox from someone in my laboratory. And they didn't even know that I had just experienced what I just did. And they said, Dennis, I think you need to know there are 3 types of human cloning. There's natural, which these 2 wonderful young ladies named Savannah and Natalie Laywell are the natural cloning method. They are identical twins who were born exactly at this time. There was reproductive cloning, which everybody on the planet wants to make illegal because there shouldn't be 2 of anybody. At least my wife says if I do that, she's going to be real mad at me. Therapeutic cloning is where we generate embryonic stem cells from blastocysts, but now we don't really have to do that because we have something called induced pluripotent stem cell technology. So we don't really have to worry about this, but they sent me this slide because, Dennis, you need to know that there's also cloning going on in your laboratory. And I said, if I ever ran into two kids that looked like that, since it was me, I'd be terribly, very sad. Okay, so stem cell biology, the message from this first part of the talk, gets people into some strange situations, but the bottom line is that we all need to be extremely interested for the reasons now I'm about to tell you. So stem cells are good guys. They live along the brain marrow of the ventricles, they live in the temporal lobe, and many, many cells throughout our nervous system that have the characteristics of a cell we call an astrocyte are actually vestigial stem cells that are waiting to be told, why don't you fix something? So I'm going to talk to you about all those guys. But I'm also going to talk to you about when you have a stem cell pathology. In the case of neurodegenerative diseases, and this is a cartoon of Huntington's disease, but it could also be Parkinson's disease, where the basal ganglia are very, very compromised. That is a potential stem cell pathology, some data I'll be talking about in a minute. And so is cancer. But we're probably born through our embryonic stem cells that build us with all of the possibilities to have all of our diseases be stem cell pathologies, because a stem cell's job is to fix us when we're broken. That's what they do for a living. If they don't, it means they're diseased. And I'm about to tell you that stem cells can contract diseases and are therefore amenable to therapeutics that target those. And the ones we're studying are the ones we eat. But I use different models that I'm going to go over very quickly, including single cell analysis, looking at markers of whether a disease or a transplant or an indigenous stem cell is happy. And then last but not least, creating a patient avatar where we can create the disease and create the immune system of that patient in a mouse avatar. So we can test our frontline nutrient and dietary therapeutics with the idea being that we're focusing on inflammation, not just because it's trendy, but because it's involved in all of these diseases you see right here. So coming back to the beginning of why we got into this and how we got into this, extracellular matrix molecules, because they're so special, allowed Katrin Goetz, who's now a very, very successful psychiatrist, investigator in Germany, to tell us in the lab that if we plate extracellular matrix molecules onto surfaces of substrates to grow mouse embryonic stem cells, when we plate the embryonic mouse stem cells onto different surfaces of proteoglycans, extracellular matrix molecules that have names like tenacin, certain glycoproteins that are developmentally regulated in those families, depending on which mixture of these substrate molecules we coat the plates with, we make different types of brain cells. So we didn't know that extracellular matrix molecules actually determine fate of cells that are growing. Using, again, these tricks of what were the molecules that make the indigenous mouse subependymal zone cells or these whisker barrel cells want to grow and be plastic in response to information from the periphery in our world. Bjorn Scheffler, who's now running his own group at Life and Brain Institute in Bonn and is a very, very gifted neuropathologist, developed a method whereby we can take any brain cell, put it on a culture dish, subject it to growth conditions, and we get stem cells, differentiated neurons, and astrocytes that we can then go back into the brain using markers we see in the in vitro cultures to see where they live in the adult brain. And that was very, very powerful. That gave us an in vitro method whereby we could grow cells under particular conditions and characterize them. So Oleg Suslov, a young investigator in my lab, determined that we have to be able to do single cell profiling because these cells are so dynamic. When we plate them on a certain substrate, they change from a stem cell to a progenitor cell to an astrocyte very quickly, or to a neuron very quickly. That if we want to feed it a particular food or drug and turn it into a better neuron or a better astrocyte, we're going to have to be able to profile these guys in real time. So Oleg, using our cell culture and our live cell systems, figured out a way in which we can do single cell profiling, actually patching onto the cell that we record from, identify it, suck out its nucleic acids or whatever else we want. And he developed a RNA expansion protocol that lets us profile cells from now until forever with very, very, very small amounts of template. Using those technologies, a former student of mine who's now in industry named Noah Walton figured out that we could find out if other brain cells in the human brain have the ability to be stem cells other than these ones that live in brain marrow. So what Noah was doing was pulling out individual astrocytes out of patients who were willing to give us some of their tissue for some neurological surgery procedure they were doing. We were also able to get them from post-mortem brains from people who've been up, unfortunately no longer with us for up to 3 days, as long as their brains are kept somewhere cold. But we're also able to pull them off of electrodes of people having deep brain stimulation and having electrodes discarded. And we could take small numbers of cells, profile them, and what Noah discovered is that every astrocyte that lives in our cerebral cortex of all of us while we're sitting in this room has the ability to be de-differentiated in cell culture while not becoming immortalized, having a normal karyotype, having normal cell cycle checkpoint genes. But one of these astrocytes makes enough neurons and glia in our culture paradigms to rebuild the brain of that patient we took that one cell from and 50 million other people. They're extremely dynamic. They're normal. When we put them into brains of immunocompromised mice, these green guys here are pyramidal cells of a human nature now sitting within the cerebrum of a mouse. They are extremely amenable to be isolated, studied, and fed good things to be able to hopefully help treat all our diseases. Now, that's the good side. The good side being maybe all of our stem and progenitor cells in the brain should sort of be considered Dr. Jekyll, in that they're good, they want to help us, they want to help diseases, but what about Mr. Hyde? Can Dr. Jekyll become Mr. Hyde? Well, the answer is yes. While these cells are dividing, what Noah found in a really interesting paper was as they're dividing really rapidly and in great numbers, they actually go into a state of aneuploidy on their way to becoming a differentiated neuron or glia. And we think the same thing happens in the brains of us in vivo, where so-called cancer stem cells have the potential to make too much tissue. So what is the definition of too much tissue in the brain, and how can we study that? So in the year 2000, Valery Kukakov and Tanya Ignatova in my lab isolated the first solid tissue cancer stem cell from a type of cancer that's cartoon-wise depicted as an avalanche here, but it's a type of cancer called glioblastoma. So again, using the same extracellular matrix growth condition assays where cells like to grow on matrix, and we took them out of cancer, different types of cancers, not just gliomas. We also studied actually osteosarcoma, colon cancer, breast cancer. They all had cancer stem cells, and many groups were publishing great papers on that. But the glioma stem cell that Tanya's technology let us discover led to us having to develop a new assay where we could do rapid screening of patients on a personalized level, where we could get cells out of their tumors very quickly, test frontline standard of care therapies, and then begin to test our new nutrient and dietary approaches. So Bjorn Scheffler, again, developed a new approach where we coat culture plates with a very special substrate of extracellular matrix molecules. He takes the specimen that the neurosurgeon gives him, he plates it onto these surfaces. We characterize them in neurosphere cultures. We generate single cell and other GWAS and omics profiling such that every patient tumor is different. And then the next job we have to do before we want to test therapeutics is we don't want to just test them in cell culture assays. We want to test them in animal models. So we have begun creating avatar mice using, right now, the non-skid gamma mouse, which when we transplant cells, because these mice don't have natural killer cells, they have very poor immune systems, they don't have microglia from the mouse in any substantial or reasonable numbers, we began transplanting just the cells into these mice that we felt were the bad guys, the truly nasty brain tumor cancer stem cell. And then we deferred to our neuroradiology and neuro-oncology collaborators, showed them the tumors in the brains of these mice imaged under very big MRIs, in this case 17.6 Tesla MRIs, where the tumor was transplanted from this patient with a glioblastoma. We had the mouse, which acted exactly like the patient behaviorally, with freezing, paralysis, ataxia, so it looked like a good avatar. But our radiology and oncology collaborators told us this is the brain of the patient. We saw diencephalic butterflies, things that you see in xenografted mouse models very not so often with other transplant approaches. So we had a good avatar. We had a really good brain cancer avatar. And Dan Silver and Florian Ziebsenrubel graduate student and a postdoc in the lab decided that they would want to study these guys for a really long time because they could now model disease in ways that was actually a dream come true. So I'm going to show you some of Dan's data very quickly, but I have to tell you that Florian came upon a way in which we could actually discover the cells that cause metastatic disease in glioblastoma. That's the green guys. The orange guys are the ones that contribute to the growth of the tumor bulk in a patient with this type of glioma. The green guys are the ones that would cause recurrent disease that we have to target. And he actually discovered a transcription factor by doing these avatar and in vitro approaches that we now know is a target of hopefully some of our new therapies that I'll be mentioning in a second to prevent glioblastoma invasion. What Dan's experiment showed was that in our avatars, we could model non-invasive low-grade gliomas where the extracellular matrix molecules actually deterred the glioma cells from migrating all over the brain and causing recurrent disease. Versus in invasive glioblastoma, the tumors stay very confined and don't have extracellular matrix to keep the cells very, very happy and proliferative, but actually confine them to the site of where the main tumor started. That is exactly what you see in metastatic cancers to the brain, particularly breast, lung, and other disease cancers that go to the brain under metastatic disease. They look like this. This would be the tumor. This would be the edge of the tumor and the normal host parenchyma. These are extracellular matrix molecules in a low-grade, non-invasive type of glioma where you don't see the cells migrating out and you see high concentrations of these developmentally regulated matrix molecules in the tumor mass. But in patients with glioblastoma doing archival analysis, we saw it looks just like our mice with glioblastoma. That is, glioblastoma is stealthy. It gets into your brain and it tells your microglia and your astrocytes, everything is fine, don't try to kill me, I'm going to migrate all over the brain, I'm going to take the brain over and eventually kill this patient. So what Dan had to do in proving this paper before he could publish it for his thesis work was that he could actually turn a non-invasive tumor into an invasive tumor. Now one would suggest you probably should have gone the opposite direction, but in this case, using digestion of the extracellular matrix using an enzyme called chondroitin sulfate ABC lyase, he could actually digest the matrix in the non-invasive and turn it in the avatars into a glioblastoma. Why was this happening and why am I really excited about the work we're going to be doing here with all of you, hopefully interested in inflammation and other things, is it seems probably the most important cell in this invasion and non-invasion story is the immune system of the brain, the microglial cell. And what Dan found in non-invasive tumors is that microglia get very, very reactive. They take on an M1, M2 versus the non-invasive type of macrophage nature, where that they prevent the tumor from growing out of its confined zone, whereas the microglia in an invasive tumor are not inflamed at all, and they just let all the cancer cells migrate all over the brain. So that provided us huge insights because we knew that microglia during development of the brain, particularly in brain marrow, make brain cells multiply, versus in the old brain, they release soluble factors in little vesicles that I'm going to tell you about in a second, called exosomes, that actually make brain cells grow processes and differentiate. So it's actually a paradox. It's two diametrically opposite functions of microglial cells in the brain that we strongly believe are related to inflammation. And that's all this slide actually says. Okay, so how are we studying this in avatar mice? We're doing it for cancer. I'm about to tell you we're doing it for neurodegenerative disease as well. Extracellular matrix molecules bind growth factors and cytokines, so pretty soon I'm going to have to start talking to you about that. The way we study that is again in vitro settings, but we also take, sorry about that, non-skid gamma mice. We take skin cells, brain cells, cancer cells, degenerative disease cells, normal hippocampal cells, anything you want. We bank those. Then we take these mice, we put in the disease they have, be it abnormal hippocampal cells in the situation of age-related cognitive decline or Alzheimer's. We can put in cancer into them. In addition, and we can put it in orthotopically or transplant them subcutaneously to do really rapid treatment protocol studies. But what we do is we want to replace the immune system of this mouse with the patient's immune system. So we were taught how to do that by great investigators at the Jackson Labs who developed this methodology where you take blood peripheral blood CD34, true hematopoietic stem cells from human being, personalized medicine type, peripheral blood, plasmapheresis or just draws. We take those cells, we inject them intracardiac into newborn non-skid mice, and up to 60 to 80% of those mice on a good day will have that mouse's immune system humanized by the patient. And then we put in the cancer, neurodegenerative disease, or whatever, and then look for their microglia interacting with the at-risk cells. I've told you about how we do that for cancer. I'm going to rapidly tell you how we do this for neurodegenerative diseases like Parkinson's disease, where we can actually take sections of mouse and human brains, incorporate them into organotypic culture settings, where in this case it's a normal a striatum of a normal mouse or a normal non-Parkinsonian patient, or in this case, a patient who had Parkinson's disease. We transplant cells, we record from the cells, we do single cell profiling, and then we feed these animals drugs. Now, why is that relevant to a biomarker I'm about to tell you about we use to analyze our avatars and our cell cultures good models of patient disease? If we're going to be giving them a phytonutrient, if we're going to be changing their diets because we believe inflammation underlies pretty much all the bad stuff I'm about to close with right now, why is this relevant to something like Parkinson's disease, where I'm going to be talking to you about exosomes in a second, which a minute ago I told you are wonderful readouts. These are little vesicles that every cell releases out into its environment when it's happy. But when it's sad and it has a disease, it releases a lot of these little vesicles, just like synaptic vesicles. And inside of them, we have proteins, RNAs, long non-codings, microRNAs, you name it. We even have human endogenous retrovirus sequences, DNA, all of the things that shouldn't be transporting from one cell to another. And what I'm about to tell you is that we are now strong believers that most, if not many aspects of neurodegenerative diseases like Parkinson's disease and Alzheimer's disease are infectious, prion-like diseases. Now, you haven't heard that just from me. Everyone knows about prions and Stanley Prusner's disciples and Jakob Creutzfeldt disease and Alzheimer's disease. But I'm now telling you we've stumbled upon something by studying our cultures and avatars that we think is involved in disease transmission. And it relates to the patients who have had fetal dopamine neuron transplants, the cell at risk for early death in Parkinson's, fetal dopamine neuron transplants from fetal donors, sometimes multiple donors to do a single patient. And when these patients presented in the clinic years later, and ended up their dopamine neurons that came from the fetal transplants had all contracted Parkinson's disease. Now, how can you explain that? When we want to study the wonderful omics approaches that Jose uses for figuring out when cells are genomically normal or abnormal, we should understand that these diseases should be intrinsic, indigenous within the cells of the brain. Here we have cells coming from donors that don't have Parkinson's disease, that in these patients that were not doing so well, perhaps decades following the transplant, but originally presented extremely well, how did they get Parkinson's disease? Well, it ends up their substantia nigra dopamine neurons compared to dopamine neurons that we can generate from patients' cerebral cortex who have Parkinson's disease are all sick. They all have a stem cell pathology. So how are we going to study this for now treatment approaches? And how can I relate this to the major goals and functions of the Neuroscience and Aging Lab here, where we're supposed to be trying to treat age-related cognitive decline, where Alzheimer's disease is only one example of age-related cognitive decline. And the way in which we're going to do that, I have to show you that our imaging data that we do on stroke foreshadows us to be able to study this in a really nice precision medicine way. So again, I'm not trying to introduce too many diseases, but Tong Zhang in our lab, in our group, has taken pediatric and adult mice that she gives strokes to and transplanted stem cells that have their stem cells filled with a quantum dot nanoparticle that is coated with gadolinium. So we can see these guys under imaging. So we transplant our stem cells in mice that have diseases. We pop them under huge MRIs, and we can actually watch stem cells almost migrating to areas where they're needed. So in real time, under imaging like that. So that's real huge, because according to this cartoon from Uli Lindvall, who discovered when we have a stroke, our actual stem cells along these brain marrow regions in the subependymal zone in the hippocampus actually migrate to the region where the stroke is, and they turn into new neurons in glia. So we have to tap this endogenous neurogenesis that exists in our forebrains all the way actually down to our spinal cord, and especially in the hippocampus, where each one of these green cells in a transgenic mouse that Grisha Nikolaukov's group made for us to study with him. Grisha's at Cold Spring Harbor and made a wonderful nestin stem progenitor cell mouse where we can actually watch these cells under various imaging protocols responding to injury and disease. And in an experiment that's kind of near and dear to my heart, and I know is pretty important to Dr. Shukat Hale as well, our NASA study where we showed these cells are extremely susceptible to cosmic radiation, where astronauts, if they would go to the moon or Mars, it ends up surprisingly, these stem cells in their hippocampus will be dead up to about 65% of the total population. So that's not a good thing. So what we want to do is we want to tap these cells to be able to have the endogenous neural stem progenitor systems that live in our brain be targets of all of the right standard of care, integrated medicine, and all other therapies to make it be that we can begin to attack diseases where we have not enough neural cells, in the case of degenerative disease, or too many neural cells, in the case of cancer. So as part of our OSCAR project, which we're really excited about, we're going to begin to study the hypothalamus, where we know from published papers there are stem cells that live along the third ventricle down at the base of our diencephalon in the hypothalamus that are very, very sensitive to inflammatory cues, particularly cytokine growth factor type messages. And these guys are in touch with systemic aging in organs such that the brain tells the body to age, the body tells the brain to age, and we think we can intervene, again, through therapies about to close with now, that target the iPS cells and the other stem cells we transplant in all of our avatars to really begin testing this now. Now, before I tell you in the last few slides what are the therapies we're most excited about, again, I just have to tell you it's worth reading about these little exosomes, 30 to 100 nanometers in diameter, that you can see under the electron microscope when you harvest them using technologies that Zandra Breakfield's lab at Harvard MGH has taught us how to do, and her former student Johan Skog, who's running exosome diagnostics science in Cambridge up the road here. We can actually pull these things out of CSF. We can show that they're filled with the abnormal proteins in Alzheimer's disease and Parkinson's disease. For Parkinson's disease, alpha-synuclein tends to be the prion protein particle for Parkinson's, that exosomes probably were released along with free synuclein in those patients who had had transplants and had contracted disease. It wasn't just they got the disease from some external exposure. They got the disease from their own disease cells releasing exosomes and exposing these cells to, in this case, it's not the prion protein. It could be alpha-synuclein, which is abnormal protein in Parkinson's. In Alzheimer's, it would be tau and amyloid that would be the conveyor through exosomes. We can talk, if you all like, another time about how Kate Candelario and I have modeled how you get exposed to these prion-like exosomes through probably viral infections through the nose and mouth. That's a crazy idea. Please don't hold it to me right now. That'll be the topic of another day. But for why I'm really excited about being a part of the HNRCA and how I'm going to have to ask all of you to please mentor me in the world of nutrient and dietary therapies. I've been working with Brent Reynolds in his lab and coming up with ways in which, from our single cell analysis, it was very, very clear that a lot of the current standard of care therapies target single molecules. In particular, everyone knows kinases. So whatever cancer, whatever degenerative disease, or whatever, be it either an inflammatory sort of oriented disease, if you take standard of care therapies, they're wonderful. And we do have some home runs. I think Gleevec is a wonderful home run. But how many Gleevecs have we gleaned so far? Not enough. So what Brent and his school of thought has taught my lab, and now my school of thought, is that we want to activate genes vertically within a pathway by targeting many nodes that are involved, in this case, in anti or bad inflammation. But we also want compounds, phytotherapeutics in particular, that will do horizontal inhibition of the inflammatory pathways and cross several oncogenic networks, not just one or two at a time. And we think functional foods are the way to do it. And our data to date so far in looking at cell culture in avatar mice, in particular looking at cancer, but we are beginning to get data on degenerative disease and hopefully age-related cognitive decline as well, is that when we have patients and mice running in tandem, and the patients are being given compassionate use compound, the same one we're giving our mice, because these are 3 FDA-approved phytotherapeutics, one from curcumin that's been nanoized to have longer biological activity, EGCG from green tea, and we stole that from the wonderful work of Samin and her lab, as well as the curcuminoids, which again we borrowed or stole from Mosen's work on the curcumins and many others. We are targeting inflammatory pathways that appear in this study to not only accompany the disease, but these are patients who had particular chemotherapies that through our cytokine arrays and our single and other cell profiling, we found out that chemotherapy put them into a cytokine storm, not just their disease. So when we gave them our triple therapeutic, phytotherapeutic, which we call epidifurfane, in combination with what we call Cancerna, which is medium chain triglycerides, basically trying to get them to be not just on a South Beach or an Atkins-like diet, but pretty much be ketogenic, is that all of their bad cytokines went down compared to standard of care only therapy. And with Kaplan-Meier curves for their cancers in avatar mice, their survivals were just absolutely spectacular. These were in combination with standard of care. Please don't get me wrong. I'm not saying we should feed people particular plant-derived therapeutics or put them on low-carb, mostly related to the Warburg effect, which cancer cells like. Just low-carb, high sort of towards ketosis sort of physiognomy, because that just seems to help. But it has to be in conjunction with standard of care oncology or neurodegenerative disease. Now, I know we work in an institution that very much favors prevention over therapy, and I'm on that bandwagon, I promise you, such that when we give mice who have cancer, Cancerna, not only does their cancer not appear as quickly when we give it to them, and in fact, sometimes they don't get it at all, but please don't let me say it's preventative just yet. But what we did notice was that when we looked at their neurogenesis in the hippocampus, the part of the brain in patients who have compromised memory have less stem and progenitor cells compared to patients who have good memory under particular memory tests in a paradigm where we actually were studying patients receiving hippocampal resections for intractable epilepsy. We found that we could actually produce more neurons in glia by feeding them this same phyto and dietary component. In conclusion, sorry for all that weird stuff in too rapid pace. I hope that what I could do was have everyone here understand things that I would love to have your help with. I have to thank a lot of people who are on this list, but right now especially the Neuroscience and Aging Lab and great collaborations we're doing to begin to conquer the neurodegenerative diseases and particularly memory-associated ones. But I would be completely wrong if I hadn't emphasized that food and the types of therapies we're studying have to be done in conjunction with standard of care things put into our brains, our mouths, and our arms. And in addition, rehabilitation, and as I tell audiences that have PT, OT, or any type of therapist in it, if you don't do brain rebuilding and brain protection while you're doing rehabilitation, it ain't gonna work. You need really good therapy. And what we've done to begin to study that is a clinical trial we did recently at a retirement community where we have patients where we're going to want to be giving them the right diets that we all know here are right. But we're also feeding their brain not just luminosity type things, but targeted behavioral cognitive testing that Tammy and others here know how to do with humans. In addition to physical exercise, where some of our patients and some of the people in this trial were not able to do treadmill or walking, and we had them do We Fit. And a very, interesting paradigm where the combination of potentially diet, but standard of care therapy, or We Fit plus brain teasing using the brain fitness program actually improved their cognitive skills remarkably. So take home message, food as a therapy, behavioral enhancement, and the right cells. Thank you. Hi, Joel.
Dennis, that was wonderful.
Thank you.
When you give your phytonutrient cocktail, to what extent do those phytonutrients or their metabolites, to what extent are they capable of entering the central nervous system? And, or alternatively, is the working hypothesis that they reduce systemic inflammation, and by doing that, secondarily reduce inflammation in the brain?
So what we've needed until now is questions from people like you to help us with that. So I can give you the bad answer. The bad answer is we chose specifically these three based on a very strong literature and our own molecular fingerprinting or biographing of cells to pick three cells or three phytos that not only cross the barrier, but are all FDA approved already. Not in the combination we chose, but for other things as FDA 1 or 2. So we did a bit of in silico design to want them to be the right ones. And then our cell culture, our mouse, and our human PK and metabolomic studies that are very rudimentary right now tell us they look good, they don't seem to cause weird things in livers and such. Even though the curcumin is nanoized for a very long biological activity, we have noticed a side effect that one of our sort of testees, who's a very close loved one of us, noticed that their anticoagulation therapy for heart arrhythmia seemed to be enhanced by our compound because of the curcumin and because of it being so bioactive because it's nanoized. They all look pretty safe, Joel, but we need to do better PK. We need to do all of the omics. I think this is a place we're going to be doing it. But right now, all things point to they get to the barrier, across the barrier. In the case of brain cancer, the barrier is compromised anyway, so they're going to get there. But in the case of neurodegenerative disease, which is our focus of work we're doing here, I think we're just beginning. And we're going to have to do this all really well and put it up against other compounds. So we're putting it up against the berries, the nuts, and what we think we're going to be having to do is have a dietary plan and a nutrient plan with all of the right readouts.
Having heard, I think, all 5 of the James Joseph Memorial Lectures, I think I can speak for all of us to say how pleased he will be, and we all are, with this exciting level of science. A couple of observations. It seems to me that the notion of inflammation is a double-edged sword, if you will.
Yep.
Which can be seen, systemically as well as in the brain, is a very interesting focus here. And I'm wondering, one question is, do you think that when we talk about glial inflammation and so forth, that we're really that what's going on in the brain is really a parallel with systemic inflammation, or is it sui generis? That's one question. And another is, I'm absolutely fascinated by the extension of the concept of the glial scar, and wonder whether, you may have heard it here for the first time, the way in which high doses of folic acid prevent neural tube defects is by creating
Interesting.
and initiating a number of events like the glial scar, which cause the neural tube defect.
Wow. Heavy. Okay, those are 2 very tough questions, Dr. Rosenberg. So I was prepared for the first one because you braced me for it by sending me emails saying, hey kid, if you think you understand inflammation and oxidative stress, guess what? So I now think about that, Irv, because I think like the yin and the yang of this should be that there's good inflammation and there's bad inflammation, and maybe the same thing with oxidative stress that I will learn from the wonderful folks here. So I now write my grants by saying, I'm going to look at M1, M2 macrophage-like profiles, that there have to be good pro and bad pro-inflammatories, and thank you for telling me that I knew nothing about that. Regarding the glia, though, I came prepared with one paper that I carry around a whole lot because it addresses exactly what you're talking with about glia and inflammation. So it's a paper published in the last couple of years dealing with a cerebrofudosis, or one of those lipofuscinosis, I can't spell that, where it ends up that one of the cerebral manifestations of that metabolic disease is having astrocytes in a highly dysregulated inflammatory state that contributes to the CNS manifestations of that pediatric metabolic disorder. And so they gave the model, these mice, a designer anti-inflammatory that they created and pretty much reversed it. So that tells me that glia and inflammation have to be studied, but there's going to be a good and a bad side to it. Now, the second question again, neurotube and folic acid.
Prevention of neurotube.
Thank you. This age-related cognitive decline will get better with work we do here. So that question, so it's interesting that I have a very good friend and colleague at Case Western who studies the glial scar from a very interesting perspective. He says it's got the good things in it and it's got the bad things. What the glial scar was invented for by a great designer was not to prevent regeneration in the case of people who have spinal cord injuries, but to actually heal the wound. So all these developmentally regulated molecules like extracellular matrix get upregulated not to prevent axons from crossing across there so you don't lose functions of limbs and all. It was to keep the place free of bugs, create a sort of cytokine storm of sorts to fight things. But then how do we get that to go away at the right time to get regeneration? So he studies this from the perspective of you got to turn on the good things and the bad things at the right time. And my guess would be, and I think you're not going to disagree, with neural tube defects or when you introduce the folates, the Bs, all of the ones that are so important, all of the ones that all of us study, including you, Dr. Booth, they have to be turned on at the right time, and they should probably be turned off at the right time. That's all I know.
In the interest of time, I'm going to ask, there's a lunch with Dr. Steindler with trainees, but anybody else who'd like to have their questions answered, I would encourage you to join. The lunch is on the second floor. I want to thank you again for a wonderful.
Thanks, Sarah.